Morning, everybody.
Good morning.
Welcome to Popular Science Lecture Series of Tamil Nadu Science Forum. This is the 77th. Every month we are conducting 2 lectures here in this series. So, to introduce about the Tamil Nadu Science Forum, I invite Mr. Sudhakar, who is the Secretary of Tamil Nadu Science Forum, Chennai South.
தமிழ்ல பேசலாமா? ஒன்றும் தமிழ்நாடு, என்னோட பேர் சுதாகர். தமிழ்நாடு அறிவியல் இயக்கம் வந்து 1983 சென்னை 81 சார் வந்து கல்லூரி பேராசிரியராக இருக்கிற நாடகத்தில் சென்னை பல்கலைக்கழகத்தை ஒட்டி இருக்கக்கூடிய பேராசிரியர்களுக்காக வந்து ஆரம்பிக்கப்பட்ட அறிவியல் பாடப்புத்தகத்துல படிக்கிற விஷயம் தாண்டி நிறைய அறிவியல் இருக்கு, அதெல்லாம் வந்து கற்றுக்கணும் அப்படின்ற ஒரு அடிப்படையில் 81 ஆரம்பிக்கப்பட்டது. முக்கியமான நோக்கம் என்னன்னா, அறிவியல் மனப்பான்மையை வந்து வளர்க்க வைக்கணும். அறிவியல் மனப்பான்மை எல்லாருக்கும் வந்து அறிவியல் மனப்பான்மையோட யோசிக்கணும் அப்படின்ற ஒரு விஷயத்தோட ஆரம்பிக்கப்பட்டுச்சு. அதுக்கப்புறம் வந்து கல்லூரி பேராசிரியர்களுக்கு மட்டும் இல்லை, அறிவியல் என்பது வந்து எல்லா மக்களுக்கும் போய் சேரணும், எல்லா மக்களையும் வந்து உள்ளிட்ட ஒரு அமைப்பா இருக்கணும் அப்படின்னு சொல்லிட்டு அது வந்து விஸ்தரிக்கப்பட்டது. அதுக்கப்புறம் வந்து சாதாரண எளிய மக்கள் விவசாய மக்கள் இருந்து சயின்டிஸ்ட் வரைக்கும் இருக்கக்கூடிய அனைத்து வகையான மக்களையும் ஒன்றிணைத்து இந்த தமிழ்நாடு அறிவியல் இயக்கம் வந்து ஒரு பேர் இயக்கமா வந்து உருவாச்சு. அதுல ரொம்ப குறிப்பா சொல்லணும்னு சொன்னாக்கா, 90களில் ஆரம்பிக்கப்பட்ட அறிவியல் இயக்கம் யாரும் கேள்விப்பட்டிருக்கீங்களா? எழுத்தறிவியல். தமிழ்நாடு மூளை முடுக்கங்கும் வந்து எழுத படிக்க தெரியாதவங்களே வாசிக்க வச்ச ஒரு அமைப்பு அப்படின்னு சொல்லலாம் தமிழ்நாடு அறிவியல் இயக்கத்தை. தமிழ்நாடு அறிவியலும் அந்த மாதிரியான ஒரு முக்கியமான பணிகளை அதுக்கப்புறம் கொண்டு போனாங்க தமிழகம். அதுக்கப்புறம் வந்து நிறைய ஆசிரியர்களை எங்களால் வந்து ரீச் பண்ண முடியுது. அது மூலியமா நிறைய ஆசிரியர்கள், நிறைய மக்களை தொடர்பு கொள்ள முடியுது. இப்போ தமிழ்நாடு அறிவியல் இயக்கம் வந்து தமிழ்நாடு அறிவியல் இயக்கம் பொறுத்த வரைக்கும் இப்போ வந்து ஒரு 5 விதமான ஒரு முக்கியமான பணிகள்ல நம்ம வேலை செஞ்சுட்டு இருக்கோம். அதில் ஒன்று வந்து அறிவியல் பிரச்சாரம், இன்னொன்று வந்து கல்வி, அதுக்கப்புறம் வந்து ஆரோக்கியம், சுகாதாரம், அதுக்கப்புறம் வந்து அறிவியல் வெளியீடுகள்னு சொல்லி புத்தகங்கள் மூலயமா வந்து அறிவியல் பிரச்சாரம் பண்றது, துடிர்லங்கள்னு சொல்லி குழந்தைகளை கனெக்ட் பண்றதுக்காக ஒரு, அப்புறம் சூழலியல்ல ஒர்க் பண்ணியிருக்கோம். என்விரான்மெண்டல் சூழலியல் இந்த டொமைன்ஸ்ல வந்து தமிழ்நாடு அறிவியல் தொடர்ச்சியா வந்து ஒர்க் பண்ணிட்டு இருக்கு. இதை ரொம்ப முக்கியமா சொல்லணும்னு சொன்னாக்கா, அறிவியல் பிரச்சாரம் தான் எங்களோட பிரதான பணி. அறிவியல் பிரச்சாரங்கள்ல ரொம்ப முக்கியமா சொல்லணும்னு சொன்னாக்கா, இது மாதிரியான சொற்பொழிவுகள், அறிவியல் சொற்பொழிவுகள் வந்து நடத்துறது. குழந்தைகளுக்கான நிறைய நிகழ்ச்சிகள் நடத்திட்டு இருக்கோம். அரசாங்கத்தோடு சேர்ந்து ஒர்க் பண்ணிட்டு இருக்கோம். தேசிய குழந்தைகள் அறிவியல் மாநாடுன்னு ஒரு நிகழ்ச்சி, இளம் விஞ்ஞானிகளை உருவாக்கக்கூடிய, குழந்தை விஞ்ஞானிகளை உருவாக்கக்கூடிய ஒரு நிகழ்ச்சி. அதை ஒரு 31 ஆண்டுகளா தமிழ்நாடு அறிவியல் இயக்கம் தமிழ்நாட்டில் வந்து ஒருங்கிணைச்சிட்டு இருக்கு, மத்திய அரசாங்கம். அப்புறம் வந்து வானவியல் மன்றம்னு சொல்லி இப்போ ஒரு நிகழ்ச்சி போயிட்டு இருக்கு. அது வந்து தமிழகத்தில் இருக்கக்கூடிய பள்ளிகளில் ஸ்டெம் ஆக்டிவிட்டியை வந்து ஸ்டெம் ஆக்டிவிட்டி மூலியமா அவங்கள வந்து ரொம்ப ஈஸியா வந்து அவங்களோட கற்றுக்கக்கூடிய திறனை வந்து ரொம்ப ஈஸியா வந்து வளர்த்துட்டு இருக்காங்க. ஸ்டெம்னு சொல்லி ஒரு மாணவியல் மன்றம், அது தமிழ்நாடு அறிவியல் இயக்கம் தான் வந்து தமிழ் மக்கள் இருக்கக்கூடிய அரசு பள்ளிகளில் கொண்டு இருக்கு. ரொம்ப முக்கியமா அறிவியல் பிரச்சாரங்கள்ல ஒரு மேகசின் நடத்திட்டு இருக்கோம். அது வந்து துளிர் அப்படின்னு சொல்லி ஒரு அறிவியல் மேகசின். துளிர்ல குழந்தைகளுக்கான அறிவியல் கட்டுரைகள், எளிய அறிவியல் கட்டுரைகள் வந்து ஐஐடியில் ஐஎம்எஸ்ல இருக்கக்கூடிய பேராசிரியர்கள், விஞ்ஞானிகள் இருக்கக்கூடிய அறிவியல் கட்டுரைகள் குழந்தைகளுக்கு கிட்டத்தட்ட வந்து ஒரு 40,000 புத்தகங்கள் வந்து தமிழகம் முழுக்க வந்து குழந்தைகளுக்கு போயிட்டு இருக்கு. அதுக்கப்புறம் வந்து துளிர் டேலண்ட் டெஸ்ட்னு ஒரு நிகழ்ச்சி நடத்தும். ஜந்தர் மந்தர்னு சொல்லி ஆங்கிலத்துல ஒரு புத்தகம் இருக்கு, அது வந்து இங்கிலீஷ் ஆங்கில அதுவும் நடத்தும். இது மாதிரி வந்து அறிவியல் பிரச்சாரங்கள்ல பல்வேறு பணிகள் செஞ்சுட்டு இருக்கோம், தொடர்ந்து இயங்கிட்டே இருக்கோம். நாளைக்கு கூட வந்து ஒரு பேரணி ஒண்ணு எல்ஏசில அறிவியலுக்கான இந்திய பேரணி இப்போ அதுவும் நடத்திட்டு இருக்கோம். இப்போ இதெல்லாமே வந்து எங்களோட பிரதான பணிகளாகி பள்ளிகள்ல பள்ளிகள்ல தொடர்ச்சியா இயங்கிட்டு இருக்கோம். இது மாதிரியான நிகழ்ச்சி நடத்திருக்கோம். நீங்க வந்து உங்க சார்பில் நீங்க வந்து வாலண்டியர்ஸா வந்து எங்களோட சேர்ந்து ஒர்க் பண்ணுறதுனால ஒர்க் பண்ணலாம். 38 மாவட்டங்கள்ல அமைப்புகள் இருக்கு. எல்லா மாவட்டங்களையும் தொடர்ச்சியாக பள்ளி கல்லூரிகள்ல கிராமங்கள்ல தொடர்ச்சியான பணிகள்ல ஈடுபட்டு இருக்கோம்.
Ladies and gentlemen, it is my pleasure to introduce Dr. R. Ilangovan. He's an assistant professor in the Department of Endocrinology at prestigious University of Madras. He holds a Ph.D. in endocrinology and has further honed his expertise to postdoctoral research positions at renowned institutions such as University of Oklahoma Health Science Center and the University of California, Los Angeles. His extensive experience in cancer biology has equipped him with a deep understanding of the complexities of this devastating disease. Now, about the lecture, it is captioned as Hope in the Horizon, Novel Therapy for Cervical Cancer. As all of you know, cervical cancer poses a significant threat to the global health, with India witnessing a staggering growth in number of this dreaded disease. In India, cervical cancer alone claims the lives of approximately 350,000 women annually, emphasizing the urgent need for innovative treatments. Dr. Ilangovan's lecture will delve into groundbreaking therapeutic approach, targeting the WNT beta-catenin signaling pathway, which plays a crucial role in regulating cancer stem cells. By exploring this novel strategy, we may uncover new possibilities for effectively treating metastatic cervical cancer, offering hope for improved patient outcomes. With this, I hand over the mic to Dr. Ilangovan to deliver his lecture.
Thank you, sir. Is it working? Is it working, sir? Yeah. Is it okay? Is it clear? Last row, is it audible? Yeah. Respected executive committee members of Tamil Nadu Science Forum, the founder, Dr. Govindarajan sir, Dr. Murugan sir, Dr. Vijayan sir, Dr. Aditya Verma sir, and other executive committee of the Tamil Nadu Science Forum, and Anna Centenary Library staff members, especially Dr. Kamachi ma'am and assistant librarian, dignitaries, participants, students, ladies and gentlemen. Very good morning to all of you. So today I'm going to talk on Hope on the Horizon, New Therapy for Cervical Cancer. Okay, we are... Well, at the outset, yeah, sorry. At the outset, I thank the the organizer of this, popular science lecture for inviting me to deliver a talk in this August gathering. So we are in the right place, I would say, because see the RAC, Gynecology and Obstetrics, and I'm going to talk about cervical cancer. So you found the right place to talk about cervical cancer. So I'm talking on Hope on the Horizon, New Therapy for Cervical Cancer. Just to give you a brief information about cancer. Anyone, what is cancer?
Uncontrolled growth.
Wonderful, sir. Awesome. Yeah. So body system has everything under control. So whenever it is overactivation or dysfunctioning, it can lead to any disease. Simply anything for thyroid problem. Okay, if it is hypothyroidism, it's as it is. We have hyperthyroidism disorder. So everything should be kept on a check. In case of cancer, when cells start proliferating, it has a controlled cell division. So whenever there is uncontrolled growth, it can lead to hyperplasia, earlier stage. It's called like there is increase in size of the tissue or gland actually. So even if it is not treated or left, it will progress later into malignant stage. When the doctor say tumor, okay, anyone tumor means cancerous? Tumor means it may be benign or cancerous. Benign is non-cancerous. Okay, so only it is malignant, it is cancerous. So many people get lipoma, okay, kind of swollen lipoma. So it is just hyperplasia, the cells or accumulation of some fat. So it is not cancer. Okay, so cancer, for the cells, it has to undergo a lot of, it has to de-differentiate, it has to lose its differentiation, probably it becomes ugly. It has the ability to move and spread, and that's very important. Spread of abnormal cells, which causes severe morbidity and mortality. So cancer is the second leading cause of death globally. Anyone? Which is the first cause of death in the world? Yeah, cardiovascular disease. Okay, that's the number one killer, and cancer is the number two. the way no cancer is, no, I mean, the rise in cancer, one day it may, no, I mean, bypass the heart. Okay, people will do bypass in heart, and cancer is going to one day bypass the heart disease. So it is the second leading cause, nearly almost 10 million deaths. So imagine, no, that much of people are dying, no, because of cancer. And nearly 1 in 6 deaths are due to cancer. And in 2024, almost now 20 lakhs new cancer cases and about 6 lakh cell cancer deaths are projected in occur in the United States alone. Okay, so data, because if you go for any scientific, no, they have a very good record. So people have no clear projection. This is what happening every year. No, you may be have of Siegel et al. No, that's a very famous authors. They every year no gives cancer statistics. Well, so the most now common types of cancer in men, so lung cancer, prostate cancer, colorectal, stomach, and liver cancer. The most common types of cancer among women, number one is breast cancer. Okay, so colorectal cancer, lung cancer, cervical cancer, and thyroid cancer. These are the most common. No? Now people know earlier days, 20, 30 years, we don't see much of those cancer cases. Nowadays, no, wherever you see some family, relatives, members, they say, in our family, this one person has got cancer. No, it has become a very big issue actually. The cancer burden continues to grow globally, exerting tremendous physical, emotional, financial strain on the individuals, families, communities, health systems. No, it's really disrupting the whole, strategy. Cancer in India. So, cancer cases are stupendously increasing in India. And so indeed, Indian Council of Medical ICMR estimates that there will be 12% rise in cancer cases in the next, 5 years. Really, it is alarming. And breast and cervical cancer, cervix, are the most frequent, cancers among women. Okay, and India, like cervical cancer now is rising very high. And if you see this, picture, it shows the dark color intensity shows the number of incidence of cases. It's so the darker shows very high. African countries, they have high incidence of. In India, is also darker. It's better. See the shade, based on the shade, no, it is still in India has high incidence of cervical cancer. And this is the mortality. The more darker the color shows very high mortality rate still in India. A lot of mortality cases associated with cervical cancer. 5-year survival. Anyone what is 5-year survival? So when the patient diagnosed, doctor says how percentage of people will survive for 5 years from the day of diagnosis. Okay, so it varies from cancer. As long it is localized cancer, any cancer for that matters. If you take a solid tumor, so thyroid cancer or liver cancer, as long it is early stage, you can by surgery you can remove the cancer, provided you don't spill out any cells. 100% curable, most of the cases. Okay, later stage it started. Where is stage 1, stage 2, stage 4, even early stage of stage 3, most of the cancer can be cured. But it goes to stage 3 or last stage. Even though many patients, they say they are in the last stage, very sorry to say, like last stage. So fourth stage, it is dissemination of the cancer cells from, say, for example, thyroid spreads to other lymphatic tissue and other things. So liver cancer spreads to other bone and others. So breast cancer very often occupies the bone and the liver and even lungs. Okay, so they get metastasized, the word metastasized. Okay, so why I'm talking more about metastasis, because it is not the primary cancer that kills the patients. Okay, only if it starts spreading, only the metastatic cancer only kills the patients. So see the 5-year survival. This was taken a few years ago. This data. Why I'm going to compare. So if you take uterine cervix, overall stage was 67%. 5-year survival. Distance 17%. Okay, so among 100%, only 17% of the metastatic cancer will survive for 5 years. Okay. See, even the latest information, the 5-year survival rate diagnosis early stage, 91% if it is early stage. Okay. It falls to 60%, from 91 to 60%. It spreads to nearby tissues actually. Okay. But see the level, it further goes to 19%. Okay. So see, even last 5 years, not very big, change, whatever the medication. It was reported 17%, even now it's 19%. Not very big with even modalities of various treatment. So we are in the era where molecular medicine, targeted medicine, things have been, it's fascinating and it's awesome research has been conducted throughout the globe actually. But still, it's really alarming. To just give brief information before getting into the cervical cancer, most of you may know the female reproductive system contains a pair of ovaries. What is the function of ovary? Anyone?
Producing eggs.
Wonderful, sir. One is producing eggs, another important.
Releasing also, releasing.
Yeah, fantastic, sir. Hormones. Okay, it's it's a folliculogenesis. Okay, it's a process where follicles are produced, ovulation takes place. Another is steroidogenesis. Okay, it has produced steroid hormone, which is the primary steroid hormone produced by ovary. Estradiol. Okay, that's why you may be knowing, like in post-menopausal women, sudden after menopause, there will be loss in bone density, and gradually, women have high chance of getting osteoporotic related fractures when compared with men. Because men, we don't see a sudden, there is an andropause, but 60 days, slowly the androgen level declines in men. But in women, immediately after menopause is cessation of ovarian function. Once it stops, the ovarian, it cannot produce ova, it cannot produce hormones. So now, because these estradiol, they have two roles. It will increase the bone formation and it inhibits the bone resorption. Okay, it stops. There are cells called osteoclastic cells. There are mononuclear cells kind of thing. These will go and bind. It will erode the bone. So don't think like if it is eroding, it is bad. It's a normal physiology. Every day, some bone will be formed, some old bone will be resorbed or eroded by the osteoclastic cells. Okay, to keep the bone healthy. So it's normal. Throughout the year, if you take in the body trapezial bone, 25% of the bone is every year remodeled. Bone formation, bone resorption, that is erosion. That takes. Why? Our blood has to maintain particular amount of calcium level. Calcium is essential for various, for neuronal, we need blood clotting, we need bone formation, we need for everything. We need calcium. Blood calcium level is important. For that, it takes place. So this produces an important hormone, estradiol. Okay, so it has a pair of ovaries, fallopian tubes, uterus, vagina, and there is a tube-shaped structure called cervix. Okay, so today we are going to talk about cervix. And the cervix is the anatomy of cervix. So we have endocervical canal. It is endocervix and ectocervix. So ectocervix contains cells called squamous cells. Okay, whereas endocells have the granular cells, different kind of cells. Okay. So coming to the physiology, the cervix connects the body of the uterus to the vagina. I said it's tube-like structures. During menstrual cycle and pregnancy, the cervical mucus secretion is thick. Each and everything, God or nature has made everything for a particular function. Why it is thick? It stops the sperm from entering the uterus because you don't need it during the menstrual cycle or pregnancy. Okay. The thick mucus also help to protect the uterus and the upper female reproductive organs from harmful bacteria. Okay, so wonderful system. Naturally, but when the mature egg is released from the ovary, the cervical mucus becomes thinner. The thinner mucus allows sperm to pass through cervix in the uterus. That's why during the ovulation period also such changes takes place. And during childbirth, cervix role is very important. That is, it widens and dilates, allowing the baby to pass through the birth canal. So during delivery, cervix plays an important role. Coming to the cervical cancer, okay, cancer, developing in the cervix naturally, that is called cervical cancer. It's a tube-shaped structure I told, it connects the vagina and the uterus. The tumor of the cervix can grow and spread or metastasize. Spreading is called metastasis, okay, to other parts of the body, and it's a malignant tumor. It starts to develop in the endo and ectocervix, and these cells do not suddenly change into cancer. I told you it will take several years, okay, 10 to 20 years, slow, sometimes mutation in the cells. It's a very long process. Even for people with who gets oral cancer, so it will mutation or changes takes place, and it will take several years to become malignant. Okay. Instead, the normal cells of the cervix first gradually develop into precancerous changes that turn into cancer, which includes cervical intraepithelial neoplasia. Okay, that's squamous intraepithelial lesion and dysplasia. So dysplasia means dysfunctional. See, hyperplasia is number increase in the cell number. Okay, dysplasia is cell started de-differentiating. Okay, those kind of stages. All the stages before becoming malignant. So cervical cancer is divided into squamous cell carcinoma. Why it is called squamous cell carcinoma? Anyone? Yeah, because the cell type is squamous cell. Okay, in the ectocervix. The endocervix is glandular cells. So adenocarcinoma. Anyone? What is adeno? Sorry.
So it is adenovirus.
Yeah, good, but no, adeno means adenohypophysis. Wonderful. Glandular nature. Okay. So adenovirus is a kind of virus, but adeno means secretory nature. Since they are secretory glandular cells, no, they classify it as a good try, but appreciate. Yeah. Coming to the risk factors of cervical cancer, so human papilloma virus infection, that is a main risk factor. HPV infection is the #1 risk factor for cervical cancer. But other factors includes weakened immune system, herpes infection, having multiple full-time pregnancies, taking oral contraceptive pills for many years, smoking, and exposure to diethyl stilbene. Okay. The thing is, no, there are, preventable thing. HPV, it can be preventable. Smoking, it can be preventable. So cancer is, most of the cases is preventable kind. And taking oral contraceptive, that's what usually I tell in the class as well as, wherever I give talk, no. So whenever the women doesn't want to have conception, there are various ways of contraception. But many people will talk, contraceptive pills. So contraceptive pills are primarily, what is the constituent? Anyone? Steroid hormones. Okay, mostly, estradiol, progesterone combination pills actually. Okay, see, you are taking these pills to stop pregnancy. Okay, but no, body doesn't. When you take, I told the function of estradiol. Okay, it stops. It is having multiple effects in the body. So when you take as a contraceptive pill, it will get absorbed in the body, reaches blood. It reaches all the organ. Okay, it reaches breast. It reaches ovary, everywhere. So if you take long term of any steroid, it will have undesired side effect. The purpose is to prevent conception. Okay, but if you take no contraceptive pill for prevention, it may cause breast cancer, it may cause ovarian cancer, and there are cases it may also leads to cervical cancer. Okay, primarily the risk factor is the HPV infection, but studies have shown even taking long time oral contraceptives. Okay, but there are different other options. Say, well, then what will I go for? There are various barrier kind of contraception, safety period. There are various kind of contraception. Your doctors will advise you. So my general suggestion is avoid taking steroids. That's the primary thing. Modifiable risk factor, I say smoking, HPV infection, oral contraceptives, we can easily modify it or prevent it. Signs and symptoms of cervical cancer, though unusual vaginal discharge and abnormal vaginal bleeding, heavier or longer menstrual cycle, discomfort while urinating, loss of bladder control, pain during intercourse, constant fatigue. That is very common for other, constant fatigue is also for other cancer also, it's kind of the pelvic pain, unexplained weight loss, as in case of other cancer also, that's and leg pain. So these are the signs. So see, our body system is such a way, sir, please, if you can come. Such a way, like our body gives a signal whenever there is any issues, it gives a signal by giving some indication. So that's why doctor, if you go to the doctor early, you can find out what is the complication and easily it can be resolved. Early stage of any disease for that matter is easily treatable. Welcome, sir. Yeah. So that's why very important. So that's why that I really appreciate Tamil Nadu Science Forum for making this kind of meeting, so that young people, old people, all kind of people know what is, say for example, simple, if any enlargement are palpable, immediately you have to go and see the doctors. Okay, there are cases, some of my friends got palpable tumor earlier stage. If they go, 100% curable. Okay, if you ignore it, if you don't treat it, even many women have, they feel shy in like there is a lump in the breast, actually nothing in that. It's just like thyroid. It's a kind of organ. So you don't feel any inhibition. Go and consult the doctor. Early stage, it's very, very important. Early stage of diagnosis. Even people, everyone working now, even starting, like what is the hot area of research now is to find a early diagnostic marker. If you take pancreatic cancer, the survival is 3 to 6 months only. You can have people like with very, very dreadful cancer, pancreatic cancer. But the thing is, diagnosis at the very last stage. Okay, that's the problem. So it's very, very important to find, and body gives some of the signals to us. Okay, any abnormality, immediately it should be taken care. Okay. So different stages of cancer, and as I told earlier, stage 0, carcinoma in situ, early form, localized, early localized advanced, late locally advanced, and metastasis. I told you up to stage 3, earlier stage, most of the cancers are curable. Okay. After stage 3, end stage, and stage 4, usually it's very hard to cure. So it is metastasized. Okay, and it affects the organ. And say, for example, say if cell is undergoing some genetic mutation, it may leads to hyperplasia. What is hyperplasia? Increase in the number of cells. Okay, hyperplasia. Hypertrophy, anyone? I could see a lot of eminent, persons there. Hypertrophy. I could see some students are here and okay, hyperplasia, increase in the number of cells. Hypertrophy is the increase in the
size.
yeah, absolutely good size. Okay. So there are conditions like cardiac hypertrophy, instead of cells become bigger. So anything, if it's abnormal, it is going to cause disease. Hypertrophy is increase. Okay. Hyperplasia is increase the number of cells. Hypertrophy is increase the size. And dysplasia, they started de-differentiating. As long as cells, they have lot of some membrane bound enzymes, they have say adherent protein. What happens? Some will help in adhesion. Say for example, epithelial marker. Okay, epithelial, it favors attachment moving. So adhesion molecule. So sometimes there are a lot of changes happens. It's called the process called EMT. Anyone knows epithelial mesenchymal transition? Okay, the cancer cells as such it cannot travel in the blood to reach the other organ. It has to undergo a lot of changes. It's called epithelial to mesenchymal transition, EMT. Okay, then it will through the blood it reaches the other organ. It will invade there. Then it undergoes a reverse process called MET, mesenchymal to epithelial. See how smart is the cancer cells. Okay, that's how they spread to other places actually. And in situ cancer, as long as it is not at the localized, surgery, most of the time it is curable. And invasive started occupying nearby, tissues, okay, that is also early stage. Once it metastasis, it's very hard to treat or cure. Okay, different stages. So that's why I told. See, I want to say 5-year survival, in situ. Okay, 100%, curable. See, it drops as long as it goes, to the next area, invasive. And metastasis, the 5-year survival drops, significantly. And that's a problem. So cervical cancer diagnosis, health history and physical examination, Pap and test, it's called Pap smear. Very often the Pap will smear of that. And they will cervix smear and they will do HPV test. It's very common, like colposcopy, like checking it, and biopsy. If the doctor thinks there may be some, kind of de-differentiation. And staging testing, endoscopy, CT scan, magnetic resonance imaging, X-ray, and PET scan. So those are the few therapies. So standard way for any solid tumor, the first, as long it's a therapy is surgery. Surgery is the first level of. And then chemotherapy and radiotherapy, based on that, the doctors, oncologists will decide. So chemotherapy, any very commonly used chemotherapeutic drug? Platinum based. Yeah, wonderful. So cisplatin based. There are various, gemcitabine. There are a lot of drugs are available. So very commonly used cisplatin and radiation therapy. Earlier they were using gamma. Okay, so now X-ray mediated. And latest, anyone? Latest radiation therapy? Yes, sir. Yeah. You see, next to our IBMS, institution, there is a Apollo proton cancer therapy, and it is the only proton cancer therapy in whole Asia. I was told, and I was surprised. Oh, whole Asia, only one proton cancer therapy. It's really thousands of 1,000 crores. Well, and there are targeted therapies. There are various targeted therapies. It may be, using antibodies-based therapy or even targeted, things. Bevacizumab is, very commonly used to target therapy, immunotherapy that targets the VEGF. Anyone? VEGF receptor? What is VEGF? Yeah, very good. Vascular endothelial growth factors. Because in cancer, when it's growing, it leads a lot of nutrients, various growth factors, all those things. Okay, so it will form blood vessel formation, all those. This process called angiogenesis. Okay, so this is angiogenesis because for the tissue to grow or survive, you need blood supply. All the nutrients need to be taken. Okay, for that the vascular endothelial growth factor VEGF is very important. And this, it will act. Any ligand, it needs a receptor on the cells. Okay, even if you take hormones, if you take a lot of hormones produced in our body, it will be released in the bloodstream. Okay, so that's a characteristic of hormone, actually endocrine gland. Other gland, if you take they say saliva, if you take saliva will be produced, it is a tube. So they are the gland with duct or glandular gland. It will be released. But the specialty of endocrine gland is there is no duct. It's a ductless gland. It produces its secretion hormones into the bloodstream. So throughout the body, no, the hormones will be no roaming here and there. Only there are specific receptors in the target tissue. There only no this will bind actually. That is the beauty. See the I would say endocrine beauty. I'm from the Department of Endocrinology, so well, so this is the way, it's so in case of this, if you want to target, so this kind of targeted therapy, target only the VEGF receptor, so that you can prevent angiogenesis and the growth of the tumor can be stopped actually. And tisotumab vedotin is a tissue factor. It's called platelet tissue factor as well as the studies have shown it is increased in cervical cancer. So they have developed immunotherapy targeting this factor. And pembrolizumab, it's PDL1. Anyone know what is PDL1? No. Programmed death ligand. So it's become, so that is also overexpressed. So therapy. So it's not, these are the say FDA approved. Anyone what is FDA? Yeah, Food and Drug Administration, the US based. So that is only deciding everything. Okay. For everything now. So that's why people know, see, though I am a researcher, though I am talking about, targeting therapies, but I strongly support traditional system of medicine. Okay, so any medicine, you say Siddha, Ayurvedic, Unani, homeopathy, whatever it is, so it is a holistic medicine. Okay, so it will by boosting our immunity, boosting our various system, that will cure most of the disease condition. Okay, for the FDA, they don't approve those kind of combination things. They want to know what is the fine compound present in that. Okay, they have a valid reason for that also. If you have multiple, compounds, say for example, I know Phyllanthus amaris, anyone? Kiranelli in Tamil. Okay, hepatitis. Okay, so it has a lot of it. It is well used for the jaundice. Okay, hepatitis B. So 25 years ago, Professor S.P. Tyagarajan from IBM from microbiology, he has patented a compound phyllanthine. From that he has patented that actually. So the company, pharma company, they will accept only it is as a single compound kind of thing. But if you take as a product, as phyllanthus, leaf extract or anything, for anything, Cissus quadrangularis, parentine, it's very good for bone. Okay, there are compound, you can take the only compound, but it will act, but you can't have the effect of the multiple components like polyherbal drug kind of thing. But each and every therapy has its own advantages. So we can't say this is the only best treatment, this is the best. We have to choose which is the best treatment for a particular disease. Okay, the studies where some of the people who are not controlling this, they will choose another kind of medicine, conventional medicine, or people who are taking conventional medicine, if they're not treatable, they are come to the allopathy, western medicine. Okay, so we have to appreciate which is the best part from any therapy for that matter actually. So these are targeted therapies. Even with all these therapies, say advanced therapies, latest therapy, FDA approved therapies, all those things, still there is see, last 20-30 years, cancer related death, it's going on, incidence of cancer is increasing on, just like the having a lot of restaurants started booming up and fertility clinics started going up. Okay, that is also there. The food, let's food be thy medicine. Who said? Hippocrates. Okay, okay, so the food, we have totally changed unhealthy food. Okay, and that is also cause for some of the cancers as well. Okay, obesity, people started too much of weight actually. Okay, then diabetes has become one of the issues when you put too much of weight. Once you get diabetes, okay, it's a metabolic disorder, then you will get all kind of padis, nephropathy, neuropathy, retinopathy, whatever pathy you say, everything, all the comes actually. So, kind of. So in case of cancer, with all these modalities, still we are not able to cure 100% cure cancer. Anyone? What may be the reason? We are giving drugs. Okay, initially, the people say, the doctor, hey, we went to the relatives, we brought the relatives, friends to the doctor. The tumor regressed, cured. After 5 years, suddenly you hear the news, tumor relapsed, or even the patient has died. It's shocking news. So there is a concept called drug resistance, therapeutic resistance. They are becoming, cancer cells are becoming resistant. And this is because of the specialized cells called cancer stem cells. Okay, they are specialized. They are very small population of the cells. Okay, they will be hiding in the cancer tissue. Okay, very small population, say 1 to 5% among the all tissue. Okay, they will be very silent. Whenever they give the therapy, whatever therapy you take, chemotherapy, radiotherapy, therapy you give, so they will, all the actively growing cells, what are dividing, growing like aggressively, they are killed actually. Okay, even that the chemotherapy will target the actively growing cells. These are very smart cells, quiescent cells. They will be very silent. Okay, these red color cells. Okay, they are like the queen bee in beehive. Okay, தேன் கூட்டுல இருக்கிற queen bee. It's like a queen bee. Okay, there are thousands of workers bee. There are male drone. Lot of bees. How many queen bee will be there in a beehive? Only one. Okay, you kill all the workers bees, drone, everything male, everything you kill. You leave only one queen bee. What happened? Repopulate the whole beehive. Okay, so current therapies, no, they are killing all the actively proliferating cells, but they are leaving these cancer stem cells. Okay, that's why, throughout the world now people everywhere we go, no, cancer stem cells, cancer stem cells, cancer stem cells. Okay, even I am last 1 decade I am working on cancer stem cells, because if you can kill those cancer stem cells, and even with available modalities, you can 100% cure cancer. That's why people are, throughout the world working on cancer stem cells. This is called bee, I mean, honeybee or queen bee hypothesis. This is the concept they are having. Okay, how we can kill the queen bee? If you can kill the queen bee, we can destroy the whole beehives. Similarly, people are targeting this. Okay, so this is I mean, book chapter. That Gautam, it also is my PhD student actually. So now he is in University of Oklahoma. He is doing postdoctoral research. This is our book chapter on cancer stem cells. So here, see the differentiated cell, as long as so they're normal non-tumorous cells. Okay, during tumorigenesis, okay, the tumor, you see there is a small population of these cancer stem cells. Imagine like a queen bee. Okay, so whatever the current therapeutics are there, okay, it is killing all the actively growing, probably all this actively dividing cells. Okay, but it is the tumor regress, but the dormant means silent, quiescent. This cancer stem cells are still they survive, and that induce the tumor. It will repopulate the again, like the queen bee repopulate the whole beehive similarly. Okay, see, you may be remembering that the mosquito coil repellent, 30 years ago when we buy, tortoise coil, those kind of thing. With the advent of that, we grow, when we lighten the thing, the mosquitoes ran away when we started using. 1 week, 1 month after, the mosquito started flying above the coil and sitting on the coil. Okay, it's an insect. Okay, then we said, oh, okay, repellent vaporizer. Okay, liquidator, they brought it actually. When we started using, 1 month, the mosquito started running away. After 1 month, they are coming and roaming around the liquidator. Okay, when the insect can be that much smart, you think of mammalian cancer cells. How smart are they? Okay, whatever we are trying, they get kicking out. And usually these cancer stem cells, they have the mechanism. There are various transporters that will take the drug. Whenever the drug, chemotherapeutic drug, there are transporters that will take this drug inside the cells. Okay, this drug will kill the cancer cells. What these, cancer stem cells do, they will expel out the drugs. Only if the drugs get inside, they can kill it. They're very smart. They develop all the adaptive mechanism. They will kick out whatever the drugs we put in, they will kick it out. Okay, even if some enters, they have developed resistance. Okay, so these are the cancer cells as it, they don't have that, but these cancer stem cells, they have this characteristic, specialized features that that's the problem for the tumor labs. So we have targeted future Wnt beta catenin signaling. I will be talking about that. Okay, so if we can target that, targeted future therapeutics, if we can kill that cancer stem cells, if it is eliminated, we can degenerate or destroy the tumor. That's the concept. So just to throw a view idea, because Wnt. So there are various signaling pathway in the body. You take anything for that. Why I'm talking about Wnt signaling? Because it is related to cervical cancer. Okay, the Wnt, don't say it's more kind of scientific. I will put it in an easy way. So Wnt gene, why it's called Wnt gene? It's derived from the name wingless gene. Okay, in Drosophila. Anyone what is Drosophila? Fruit fly. Now we see small fly, microscopic flies. So I mean small flies, flying over the fruits. They are Drosophila, fruit flies actually. They found that when this gene is affected, no wing formation. So because it's a polarity and a segmental regulator protein actually. So it is if it is affected, this gene is affected, there is no formation of wing in those. So that's why named as wingless gene of Drosophila. Similarly, they found another gene, what INT gene, proto oncogene. It's a mouse memory oncogene, integrator complex. int. Okay. So what? No. So they found similar kind of, the gene in both, Drosophila and mouse mammalian. I mean, viral integrated int gene actually. So they made a portmanteau, just blending of words to taking this wingless and int, and they made it as a vint, like blending the words, like brunch. What is brunch? Breakfast and lunch. So similarly, they made it as a wingless int as vint. Okay. Well, this pathway, there is a signaling pathway through applied on. There are non-canonical pathway and canonical pathway. So typical canonical pathway, they take various molecules. One protein will activate another protein will activate another protein. That's how any disease condition or anything, abnormal expression or low expression of any protein leads to a disease condition. Canonical Wnt pathway, it's also known as Wnt beta-catenin pathway. Okay, so beta-catenin is a effector molecule. It's a protein. Okay, it's present in the cells. Okay, and Wnt is the ligand. As I said, like there is a ligand, it will go and bind with the receptors called Frisell receptor, then activate various intracellular proteins, and ultimately it will activate transcription factors. Transcription factors are the factors that induces transcription. Transcription is, what is transcription? Yeah, synthesis of RNA. Okay, so this. Well, this pathways, they control cell proliferation. That's the why, canonical, non-canonical pathway. Why we are talking on canonical pathway, Wnt beta-catenin pathway? Because it regulates cell proliferation, and cancer is because of uncontrolled proliferation. Okay, that's it. So major Wnt pathway, just I mean, just to have an idea on that. I mean, they may be little more scientific, but just I want to throw like major signal pathway, Wnt beta-catenin pathway, non-canonical planar cell polarity pathway, non-canonical Wnt calcium pathway. It also regulates, it has gene transcription. It is regulation of cytoskeleton and modulates calcium level. So we are going to focus on this pathway because gene transcription, because in cancer condition, lot of transcription takes place, new protein will be produced because the cancer cells start growing. They need a lot of nutrients and other various factors for the cells to grow actually. So now that we are going to talk about that. And Wnt beta-catenin signaling pathway plays very important role in proliferation of the cells, survival of the cells, differentiation, migration, the cells to migrate, and importantly self-renewal of making new copies of stem cells. Okay, many times the cells once divide, they get differentiated. Once it's differentiated, we can kill it. They have the stemness, means then only the problem comes. For health, normal health also is good. Okay, whenever we have a bone fracture, the stem cells plays an important role. Okay, they will come and they become bone cells. There will be bone marrow, stromal cells, a lot of cells are there. Okay, whenever tissue damage, we get a scratch, after 1 or 2 weeks, healed up. How? There are stem cells there. Okay, normal stem cells. They will start and growing. They produce the similar whatever the skin cells, the stem cell produce skin cells. Okay, bone cells, they produce bone cells. Okay, from the stem cells. So stem cells have the property to become any kind of cells. Okay, at least 4 or 5 lineage. Embryonic stem cells, they can become all kind of cells, but pluripotent stem cells or whatever the cells we are mesenchymal stem cells, they can give to an array of 4 or 5 lineages of cells. Okay. So Wnt beta-catenin pathway play an important role. Developmental process. That's what, if it is affected in Drosophila, even the wing formation is affected. Even in mouse and animals, they study like, because it is plays an important embryogenesis. For the formation of the embryo also, it is very much essential. What happens after expression during the embryogenesis, they will be shut down. Okay, their activity will be down whenever there is a required. But in cancer condition, problem comes when it is overactivated. Whenever no particular thing, unnecessary genes are overexpressed, that causes cancer. Not only for development process and maintenance of adult tissue homeostasis, for tissue damage, all those things, they play an important role. So this is the just schematic diagram of the Wnt signaling pathway. I told there are ligands, Wnt. Okay, there are receptors, Frisell receptor, LRP receptor, co-receptor. Okay, when there are antagonists, they are not chemical based, they are normal. They are present in the body. Okay, natural antagonist of this Wnt pathway. They go and bind and inhibits binding of Wnt with this receptor. As a result, what happens? There are various enzymes. I don't want more detail. So glycogen synthase kinase 3. What happens? This is the important protein which I am going to talk about, beta-catenin. Okay, I'm talking about Wnt beta-catenin pathway. This is the effector molecule. Okay, what happens? It will phosphorate the protein. Okay, remember in normal physiology, most of the time, say for example 99%, for the activation of the protein, phosphorylation is very important. If the protein, if it is phosphorated, becomes active. There are exceptions. Nature always has exception. This kind of protein, if it is phosphorated, it will get degraded. Phosphorylation followed by ubiquitination mediated degradation. Okay. You simply remember whenever there is no Wnt molecule, this get degraded so that no new gene expression or protein production is affected. Here, in the absence of Wnt antagonist, okay, whatever it is there, if this ligand Wnt binds with the receptor, it leads to activation of this beta catenin. It increases along with the T cell factor and LEF, T cell factor and lymphoid cell factor. It will stimulate the new mRNA expression of, naturally it leads to production of a lot of protein that will help in the cell proliferation, growth of the cells, migration, all the important proteins. So studies have shown like apparent abnormal activation of this Wnt pathway in various cancer. Adenomatous polyposis coli, it is one of the gene actually, and they found it is mutated in hereditary colon cancer cases. Okay, and studies have shown activation of beta catenin T cell factor signaling in colon cancer by mutations in beta catenin. Studies have shown this beta catenin is mutated. What happens? Mutation can be activating or inactivating. Okay, in case of here, it is activating so that it produces more protein. Loss of APC function leads to stabilization of beta catenin and formation of C. See, APC is a tumor suppressor protein. Okay, where many people may be working in science know P53 is a tumor suppressor protein. It will suppress the tumor cells or growth of the tumor actually. Okay, if it is not there, if it is mutated, it is non-functional. What happens? The cells start growing actually. Okay, this is kind of APC is a tumor suppressor. It is lost in this function so that beta catenin see, imagine beta catenin is going to cause cancer. Okay, when there is no stop. Okay, if there is no police, the place where robbery is taking place, then naturally the robber will take. So similarly, they will have a check on this oncogenic protein actually. Since because of mutation, this tumor suppressor, it can lead to cancer. Okay. So mutation activation of this proto-oncogene is a common event development of various tumors, and Wnt inhibitory factor 1 is silenced by promoter hypermethylation in various cancer, okay, and especially in cervical cancer. So we are the first. So when I was doing postdoctoral research in Dr. Lourdes Pamedo's lab in University of Oklahoma Health Science Center, so she is a wonderful mentor actually. So she is a MD pathologist doctor, then PhD. Okay, she is a wonderful human being and a great scientist. So she was mentoring me and she was telling like working on cervical cancer. Okay, we started accidentally working. We were working on salivary gland cancer because of change in cell line. Accidentally we started working on cervical cancer, and we were the first to report like this Wnt inhibitory factor 1, which is like a tumor suppressor protein. It is less in the cervical cancer. Then we tried, if we can overexpress this, give this protein, whether we can cure cancer. We did some, couple of experiments. Just I will share you. And this apparent this chronic protein means continuous activation. Okay, continuous activation is found in various tumors, and the cytoplasmic and nuclear accumulation of beta catenin is a hallmark. So when you see the cells, if it is more in the cytoplasm and nuclear, if this protein, it tells like it is going in activating stage and it is inducing transcription. Okay, see here, more than 70% of cervical cancer specimens, they are positive for this beta catenin or Wnt. It means when you take a cell, this protein is highly expressed, means it is having some role in cancer. That's how we find it. Okay. Please stop me if you are not able to follow anything. No, please stop me. Is it okay or I'm rushing actually? Yeah, please feel free to interact. Well, so human papillomavirus, I was talking, HPV plays an important role in the initiation and progression of cervical cancer. Okay, 99% no, it is because of so infection with HPV is the first hit in the cervical cancer development. Okay, first for the cervical cancer, you need to get a HPV infection. But remember, not all HPV positive cells, they progress to cancer. It's not all HPV positive. Many people will be HPV positive, but they never get, cancer. Okay, just know. Because activation of the Wnt beta catenin pathway is considered as the second hit. Okay, once there should be infection with the HPV, and in those case cells, if this beta catenin Wnt beta catenin pathway is activated, then only it can leads to the progression of cervical oncogenesis. Okay, studies have shown that. Okay, however, the precise molecular mechanism of the cervical cancer progression is really unclear or less explored. So this is the studies where we did. So we used HeLa cells. Anyone know what is HeLa cell? HeLa. Name of African lady in 1951. No, she had no cervical cancer. Okay, the doctor. Now, yesterday also in IBMS we were attending a research ethical meeting. Okay, so there are research ethics. You can't take I mean blood samples or any tissues from a person without their consent. But those days, 1951, in this African lady, removing the doctor took the cervical cancer tissue and he grew it actually. Okay, they made a cell line. So first they found cell line. And people, it's a cervical, cancer cell line actually. Hela means Henrietta. Her name is Henrietta Lacks. So they used first, two-letter Hela. So Henrietta Lacks, name of the patient. They made it Hela cell. It's very widely used cell for various to see the metabolism of the chemical, cell proliferation, differentiation, migration. For this, very commonly used cell line. For any experiment, people working with cancer, for sure they will use this cell line. It's a cervical-based cell line. So what happened? I was talking about a Wnt inhibitory factor 1. Okay. So when we transfected that, okay, you can give treatment one by giving a drug, okay, WF1 protein or inhibitor like that. What we did there, we had a plasmid, okay, it's a gene kind of therapy. The plasmid produces this Wnt inhibitor factor, tumor suppressor protein, okay. So there is a procedure, a procedure where you use, lipofetamine based, lipid based, technology where you can make this plasmid or a DNA or the gene to put inside the cells. There are strategies you can do in the lab. Anyone can do that. If I teach, anyone can do that is a technique. By doing the technique, we are putting this gene inside the cells where this gene is less or not present. Okay. When it overexpresses, it is killing the cells. This was, say for example, this is the HeLa cells without any treatment. See, it's 24, 48, 72. It's keep on growing. Okay, you can grow the cells in nutrient media. A cell culture lab, you can do that. When we overexpress this tumor suppressor protein, it is killing the cells. Okay, so it means by targeting the beta catenin pathway, it is this the tumor suppressor is killing the cells. We'll see the number of what may be the mechanism. So when the cell are killing the cells, there are various mechanism. Okay, cell cycle arrest is one of the mechanism. We found there is a G2M phase arrest. It's just cell cycle phase. It is arresting and it is that's how it's killing the cells actually. And we looked for how it is killing apoptosis. It is apoptosis is a programmed cell death. It's a mechanism by which cell can be killed actually. So we found it is inducing cell death actually. And we did finally, if you want to go for any drug development, you have to go for preclinical validation. So preclinical means animal model. Okay. So anyone, what kind of mouse is this? Anyone? Louder. Athymic nude mouse. Okay. Since there is no hair, it's called nude mouse. Okay. The skin. Okay. So they have made an athymic. Thymus is not there. We have a thymus gland, thymus, because thymus plays an important role immunorelation. So they made a transgenic mice without thymus, so that they are immunocompromised. Okay, immune system is not functioning there. Why we are making this mouse? Because we are going to inject human cells. Okay, say for example, some foreign body enters our body. Okay, what happens now? Our immune system will try to destroy it. Okay, if you use a normal mouse, okay, inject the cells, the immune system of the mouse will reject the cells. You won't be able to form tumor. Why we want to form tumor, human cells based tumor in mouse? Because to check because we can't directly go on humans and check it. So it may kill the the patients, or it may have deleterious effects, side effects. So first we have to do animal experiment. So we injected these HeLa cells. Okay, they form very big tumor. Okay, once the palpable tumors are formed, we started injecting this plasmid. Okay, the gene therapy. Like kind of we are injecting the tumor for 7 weeks. We treated. See here, the without any of this WF1 gene, it's keep on growing there. Whereas it is not. It is suppressing the tumor. So by targeting the Wnt beta catenin pathway by overexpressing tumor, so we could show like we can reduce the tumor. Okay, this is the study reality. The tumor size. See after that. So if you have given another 2 weeks of treatment, this will disappear. Okay. But why we didn't do? We need a tissue to do molecular work to show like this is a mechanism. Being a scientist, that's what we do. So this is a section from the human tissue, the tumor which I showed, the see in these cells, this human HeLa cells, they are trying to invade the muscle cells of the mouse, because we've injected in the mouse, no, the flank of the region of the mouse, no, it shows no the tumorigenic property of the cells. But here you see, it is not no like invading. Okay, it says a very distinguished border, shows tumor suppressive effect of this WF. Well, so that's what the other part of the work. So when we started no going for using a tumor suppressor protein, which is present no outside the cells, which we are injecting and doing that. Okay. Ultimately, what this WFN protein does, no, it will inhibit the beta catenin only. So then we, thinking like, why don't we directly target the beta catenin? Okay, rather than using that, no, that because this is the effector molecule. Okay. So we used two approaches. Okay, one small molecule inhibitor based. Okay, just to say it's a chemical inhibitor, potent inhibitor of catenin responsive. So it is going to affect the binding of TCF and the beta catenin. So beta catenin activity, we want to stop that. We use this thiazolidine group of inhibitor. It's a chemical based inhibitor. Another thing, we used siRNA, small interfering RNA. If you have those kind of small interfering RNA, it will go and bind, and the what is your target RNA, it will downregulate or silence it. Okay, so that no particular protein cannot be produced. That's the idea. Okay, used two approaches to stop the beta catenin expression function. Okay, so any study for the students, especially any study you are going to do, you should have a hypothesis. First you should have hypothesis. You say like, okay, the targeting the beta catenin with small molecular inhibitor of beta catenin responsive ICRT14 or small interfering RNA, siRNA, may kill the human metastatic cancer cells and reduce cancer stemness. Always no, you should have hypothesis. We are thinking, proposing it may work. Sometimes it may work or it may not work. Okay, it doesn't matter, but you should have an hypothesis. Then to test the hypothesis, we framed these objectives. Effect of this small molecule inhibitor as well as the silencer RNA on cell proliferation, migration, apoptosis. These are the part of the process that takes place during tumorigenesis. Okay, we saw all those things. Then we looked for a very molecular, so being a scientist, to see what may be the mechanism it is killing. But anything, to develop a drug, you need all molecular, validation and evidence. So cell lines, we used metastatic human, cell. There in the study, that I did in Oklahoma University, that is based on the HeLa cells. Okay, so this is a metastatic cervical cancer cells. So remember, I was telling like you can cure the actively proliferating cells, all those things. Treatment for metastatic cancer is not available. That's why our focus to, is there any way you can try to kill the metastatic cancer cells? That's why we use this metastatic human cell line ME180, and these are all the characteristic features of metastatic cells actually. So this drug was purchased from Sigma Village, and the siRNA we purchased from Santa Cruz Biotechnology. These are the chemicals we use. These are the various assays, how cells are growing. There is a strategy to associate, how to see the cell migration. There is a say you can do that. Cell population, cancer stem cells. I was telling small like queen bees, there are various technology we can, using that kit you can find how much of cells are, how much number of cells are there you can do. There are the gene expression studies. Many people working in science might be doing real-time PCR to see the mRNA expression and Western blot for protein expression, those kind of we treated actually. Just I will skim through the result actually. So when you treated this, ICRT, which is an inhibitor of beta catenin pathway, so 0 hours, no treatment, that's the time. So after treating, see decrease in the number of cells, 48 hours, 72 hours, So this drug is killing the metastatic cancer cells. Okay, these cells are hard to kill and they are killing. And this is the called scratch assay or wound healing assay. Okay, because we are inducing a wound. Okay, we have a cell culture plate where you grow the cells 80 to 90%. It will be full of cells. You take a micro pipette, the pipette which you use for the experiment. Use that pipette. Make a line with the pipette tip. It's a scratch you are making that. Okay, then give the drug and take the picture after 24 hours, 48 hours, 72 hours. See. 0 hour, see the with the this much of amount wound or this is see after 48, 72, the cancer cells they're growing, they are healing the scratch area. Okay, the wound healing. It shows the cancer cells are migrating and occupying the place. This is what happens in our body also. Okay, observe the other way. The here also the same. When it started, same size of wound is there. Here, see after that treatment. This is healed up. The wound, no, these cells are migrating very faster and occupying the place. But here, no, they are not able to. It means it is inhibiting, suppressing the migration of the cells, spreading and moving. No, it is inhibiting. So clearly, this drug is preventing the migration of the metastatic cancer cells. When we look for the apoptotic assay, this apoptotic assay is programmed cell death to see, and this is inducing, cell death. And there is a mechanism, senescence. Anyone, what is senescence? Yeah. Sorry? Yeah. So aging. Okay. So cellular senescence. Okay. If you can know the cancer cells, what happen? No, they will skip the senescence. Okay. Telomerase is one of the important enzyme. No. So whenever during division telomeres we lose telomerase, this telomerase will make it so that aging of the cells will be prevented by this. Okay. These cancer cells, they have the ability to get rid of or kick out all the mechanism protein that stops senescence or aging, because if aging, no cell will die actually. So they have ability to stop senescence or aging. But if you can induce senescence, aging in the cells, cancer cells, you can kill it actually. Okay, that's that. See, when we did blue color shows the blue color cells, these are senescent cells when compared with control or vehicle. Vehicle is the medium in which we prepare the drug. Okay, so it is increasing the number of increasing the number of cellular senescence. It means our treatment is inducing aging of the cells, cancer cells, so that it will die actually. And this is the cancer stem cell population. It's an assay called ultrafluorescence assay. You can use the kit, add the reagents, then do fluorescence activated cell sorting. It's one of the equipment you use for see the number of cells. Say for example, in a control there is about 7 to 8% cells. The like I was talking about queen bee, okay, kind of cells. See, it is drastically our treatment is decreasing actually. See, like bunches of grapes. So there is we grow in tumerosphere. There is a medium in which you can grow. The previous picture I showed are 2-dimensional, 2D. The cells will attach in the plate and grow. This is 3D. They don't attach. They form spheroids, like grapes, bunch of grapes kind of thing. They grow. So you start growing. It shows the cancer stemness, the ability to kick out the drug or for easy understanding. If there is more cancer stemness, then it will get rid of the drugs, whatever drugs. See, by giving this our drug, see, it is almost totally we couldn't find any tumerospheres, balls of this stem cells there actually. So it is a number of spheroids formed. For example, 60 spheroids we counted in whole thing. So it is drastically decreasing the number of tumerospheres formed. And this is the size, diameter, size, how much size of this. It is drastically decreased. And we looked for the molecular mechanism, mRNA expression of the various oncogenes are decreased. These are stemness marker. Okay, I was telling cancer stemness as stemness. Stemness is they can give rise to a lot of other cancer cells. Okay, if by decreasing these SOX2, now they are the cancer stem cell markers. Okay, these mRNA expression are decreased. Then we look for the protein expression. Just what I'm trying to say, these what are the compounds we use? It is decreasing the bad thing and increasing the good thing that can prevent cancer. That's what we are trying to say. So those are with the ICRT, small inhibitor. Then we did siRNA, small interfering RNA. Similarly, we did the study and we could observe same kind of decreasing the number of cells, cell proliferation. This is the viability is decreased. So here no cells about 85 to 90% viable. It had come to less than 20% viable. So it is the thing is there are studies where you can kill the the regular cancerous cells. Metastatic cancer, that's a very thing. Such a very big difference, using our drug. It's really promising. Similarly, we did the study with this siRNA, silencing RNA kind of thing. Again, see, it got healed up, but here it is not healing. So migration, the movement of the cells, it is drastically affected by it. So this is apoptotic increased. Okay, senescent cells, number of cells are increased, positive cells, and bunch of these spheroids are grapes of this shows cancer stemness. Okay, like queen bee property I was telling all about. It is our treatment is siRNA mediated. It's totally reduced or stopped the spheroid formation. So this is the number of spheroid and size of spheroid. This is the cancer stem cell population. Okay, drastically it decreased several fold. And the mRNA expression of these important factors that plays an important role in Wnt signaling as well as the cancer stemness, all got decreased by our treatment. SOX2s, SOX9, OC3, these all are, no? See, this is the normal expression after treatment, it is decreasing. Here also expression, it is decreasing. Here OCT, no? So all the cancer stemness marker, because this only leads to the cancer stemness. This only causes chemotherapeutic resistance. This only causes cancer relapse. This only causes all the cause for that. This, the pluripotent stem cell marker and our drug drop everything. Okay. Summarize. The inhibition of the beta-catenin exerted tumor suppressive effects as observed by a reduction in the cancer cell viability, migration, and stemness of the cancer cells. And also it induced apoptosis, cell death, and also senescence, the mechanism by inducing aging of the cells, it is caused. This ICRT, this is chemical inhibitor of the Wnt pathway, as well as small interfering RNA. It down-regulated the gene and protein expression of key Wnt signaling components, as well as the various markers or components of this cancer stemness that causes this cancer resistance, and while increasing the pro-apoptotic and tumor suppressor proteins. And this shows all molecular mechanisms by which targeting the beta-catenin, which ICRT14 chemical inhibitor or siRNA, by regulating viability, migratory potential, inducing apoptosis, by decreasing the cancer stemness and cancer stem cell population, EMT markers, angiogenic and inflammatory marker, tumor suppressor gene, stemness gene expression, oncogene expression, apoptotic protein expression, and regulating the anti-apoptotic protein. By regulating various bunch of signaling molecules, our treatment approach of using small chemical inhibitor or siRNA mediated knockdown killed the metastatic cervical cancer cells. So to conclude, our findings demonstrate that suppression of the beta-catenin signaling pathway using a small molecule of siRNA or siRNA reduces the proliferation and stemness of metastatic cervical cancer cells, possibly through the regulation of these critical genes. So thus our studies suggest that small molecular inhibitors or siRNA could be considered as a potential novel therapeutic agent for the treatment of metastatic cervical cancer after preclinical validation. We have to do like mouse, anthemic new mouse or xenograft tumor mouse model, and after clinical validation, it can be translated as a drug for metastatic cervical cancer. So I would like to, acknowledge various collaborators and professors, Dr. B. Ravishankar from University of Madras, head of the Department of Endocrinology, Dr. Ilangovan, Farming State, Dr. Srinivasan, Dr. Michael Arundas, Dr. Balasubramanian, Dr. Satish from SRM, Dr. Narajan from Sri Ramachandra, Dr. Northwestern University, Dr. Sridhar Muthuswamy, Karnataka University, Dr. IIT Madras. Most importantly, this work was, initiated all the brain behind is my postdoc mentor, okay, Dr. Lourdes Camilo. She's the main, everything is all because of that. When I was doing postdoc, she was, still I'm collaborating with her. And this work, my lab members, Gautam, Manju, Shabana, Agalia, and Muhammad Adil, actually. Most part of the work was done by Gautam. And this work supported by SCRB, Science and Engineering Research Board Grant Funding Agency, and special assistance by DST. First phase 2 and the UGCSAFT DSA 1 is sincerely acknowledged. This is my team, is Gautam. Okay, so Manju, Shabana Agalia, and he is Muhammad Adil. He has recently joined Ph.D. early. So Gautam is, working in the same lab where I was working in University of Oklahoma. Okay, he is working there. And Manju, she is working on how BPA, bisphenol A, the plasticizer. That's why we use BPA-free bottle, okay. But already whatever we have taken or still many, it is having drastic effect on various organ system. She is working on how it affects the bone cells. Okay. And Shabana, she is working on how histone medial transferase. Okay. So it's an epigenetic regulator. It's a role in cervical cancer. Agalia, she is working on polycystic ovarian syndrome. So how vitamins can help in, because polycystic ovarian, it has become a very big issue. On paper, we say 8 to 23% of reproductive age girl may get PCOS. But in reality, if you check, 50%, wherever you ask any, brother, wherever you ask, the girl is having PCOS. It has become a very big issue. So she is working on that. And Dr. Adil, so he is a diabetologist doctor. He has joined So we are initiating work on diabetes and neuropathy actually. So earlier Vijayan sir was asking me, so if I can give a talk in diabetes, hopefully next year, if there is enough data. So we are planning for animal study as well as clinical study on diabetes. And Gautam, he did most of the work in cervical cancer. He's an outstanding researcher and he's an amazing worker. That's why he is now in University of Oklahoma doing his postdoctoral research actually. Well, thank you so much for all your attention. I'll be happy to answer the questions. Yeah, please. Yeah, please sit down and you can ask me.
Sorry,
Please sit down. Oh,
there's any session to the work.
yeah,
Fantastic.
thank you.
My question is, what are the instruments and the technologies we have involved in the isolation and the characterization of gene expression?
So,
Based on the gene, what are the main source? And tell me about that isolation characterization process.
yeah, see, regarding gene expression, so after the treatment, we have the cells growing in the plate. So after doing the treatment, you have to harvest the cells. Okay. Is it audible for everyone? Okay. Yeah, thank you, sir. Okay, so after the treating the cells with whatever inhibitors or even siRNA, okay, you can harvest the cells. If it is for RNA, there is a reagent called trizone. Okay, you will add that. So you will take there is a procedure, as per the manufacturer's protocol. You can follow and you can get total RNA. I see the total RNA. Okay, the RNA then you have to convert to cDNA, complementary DNA. Okay, then you have the specific primers. Okay, for a gene, whatever gene, Wnt molecule or any pluripotent stem cell marker, we can do the PCR. Okay, earlier we were using a semi-quantitative PCR. Now because technology, real-time PCR, we can see increase in the what cycle, threshold cycle the gene is expressed. Based on that, you can see mRNA expression is increased or decreased. Same way, if you want to see the protein, okay, ultimately whatever RNA, sometimes the RNA may increase or decrease. It may not reflect in protein. Okay, that's very important. Is protein is the very important. Ultimately, it should end up with, of course, non-coding RNA, they play an important role in gene regulation or there actually. But functionally, the particular protein function, you have to look for protein. By Western blot, you can use a RIPA buffer. It's a radioamino A buffer. You have to use protease inhibitor cocktail so that when you lyse the cells, each and every cells, there are a lot of protease in it. It will destroy. So you have to use protease inhibitor cocktail to stop the activity of protease, to degrade whatever the protein is there. Then you have to do SDS page, polychromate gel, from the gel you have to transfer to the PVDF membrane. Okay, and you can probe with antibodies, secondary antibodies, and you have a chemiluminescent equipment. We have all the equipment in our department. So seeing that, you will see like band kind of thing. Okay, based on the density of the band, you can say the protein is high or low. Based on that, you can see. So that's what we found, like using our drug. Whatever the protein is not good, it is decreased by the treatment. Whichever is good, like tumor suppressor protein, it is increasing. That's what most of the other drugs also works actually, but they are used for other cancers, but not studies on metastatic. That's the main thing. I would like. There is no studies on metastatic cervical cancer as we have done using these compounds. Yeah. Right.
Synthesis of cell.
Mhm.
What are the drawbacks of faces? For example, because I have worked in exosomes given the cells, mesenchymal cells. The exosome population is very low.
Yeah. Okay. Can you please tell your name and introduce yourself so that everyone knows where you are working, your name?
Currently, I'm not working.
Okay, no problem. But earlier you were working.
Basically, I'm a PG biochemist.
Oh, nice.
I have worked in science and therapy, CSA, CMC.
Please sit down.
That's like I need respect.
Okay. No. Respect. No, I mean, it's not. No, it's respect.
That's fine.
No, not at all. Please.
While preparing that exosome demand from these kind of cells, we have face many drawbacks. The population of cells is really very nanometer. We cannot able to see in the naked eye by soil only %. So I'm asking you, what are the drawbacks you have faced in synthesis of gene on the protein expressions?
Yeah.
One more question I have to
ask.
Then, do you think the exosomes have any role in your cervical cancer studies? And what are liquid biopsy? So I read about the liquid biopsy. What is the role of liquid biopsy currently trends in our Tamil Nadu as well as India?
Yeah.
Thank you.
Wow, excellent questions. Actually, first number one, So my collaborator, Dr. Satish Ramlingam from Genetic Engineering Department of SRM-IST, is working on exosomes, colon cancer. So, the exosomes, like, cell-to-cell communication, now it is regulating other cells. See, some cancers are there, by producing exosomes, these exosomes are inducing the normal cells to become cancerous. He's doing outstanding research actually in exosomes. So we are planning to do, but right of now, no, we haven't done exosome studies, but he is working on exosomes. We didn't have any difficulties with, because of treating the cells, cancer cells, and collecting the RNA and protein. So we are fine. And even for the stem cell thing, we are collecting the cells. The only thing we have little problem, it's very common problem in India. Okay. So in USA, for example, nearly, 10 years I worked in US actually. The thing is, maybe 1 or 2 times I got contamination. Okay, the cell culture, when that too, when we are using like human, samples, tissue samples from that, and we grow this, that time only we have got here, because here, very often, because the cell culture, a lot of things, but air quality, a lot of things, the high chance of getting contamination. That's the only with anyone for that matter, actually, those who are working, so because the air, the quality of air, a lot of factors makes actually. So that is the only thing. Whenever we get contamination, we have to fumigate the room. Okay, we will do that, clean the room, okay, sterilize it, and we use it. That's the only thing. So that's the first part. We didn't have any difficulties, just collecting cells and doing all kind of thing. mRNA, once we get total RNA collected, you do mRNA analysis, and again, the protein lysate, once we take, we do protein. We didn't have face any difficulties. That's one. Regarding a liquid biopsy, so okay, people started so even we are planning for cervical cancer using the blood sample, looking for the diagnostic marker. Okay, so that's I was talking about diagnosis. That will be the great thing actually. We are planning to do that actually. Indeed, we planned doing cervical cancer, with Madras Medical College. So we are working on. We haven't started that actually. Couple of years later, we got the human ethical, clearance for that, but due to various reasons, we couldn't continue that. But again, we are reinitiating actually. So we are going to do on those aspects actually. Good question. Yeah, please, sir. Yeah, one second.
My question is more about the vaccination. Is it possible you can tell about it?
Yeah, absolutely, sir.
Okay, that is only for. Yeah. Yeah, good afternoon, doctor.
Good afternoon,
Is
sir.
anybody hear me?
Yes, sir, absolutely.
My question is more about the vaccination. I'm not aware, but I can pose a question, I hope. Can you throw some light on the vaccination?
Yes, sir.
Thank you.
Yeah, it's a very good question and important question, sir. With respect to HPV infection, so God has it is the approved vaccine available, HPV, protection vaccine. It should be given before the first sexual act. Okay, so maybe for the girls, at the teenage, if you can give, it will protect. That's what they claim, 99 to 100% it protects. Once they now had like sexual intercourse, then it may not work actually. So Gardasil is the, even Indian government approved HPV vaccine that protects cervical cancer. That's why, in US, the incidence has drastically decreased actually. Okay, for sure, this HPV vaccine is effective in preventing. But different people know there are different school of thoughts. People say vaccine, it may be protecting this, it may have some side effects. That's a matter of gray area. People say sometimes. Even yesterday in the ethical we were discussing. Now in a short period of time, this COVID vaccine have been approved. Okay, very short period. So because to develop any drug, it takes few years actually. But it was a pandemic. No other go. We compromised with a lot of things actually. Even I guess like Dr. Aravindan, he gave he was talking about COVID, I mean vaccine, I think so. The thing is, so always any drug will have its some side effects, but in a holistic way, see, most of them it is protective actually. So for HPV, for prevention of cervical cancer, Gardasil, which is approved vaccine, is recommended by the clinicians actually.
Part of the same question. That's a age limit. I mean,
Not age limit. So before having first, so yeah.
it's not applicable for somebody, age people, it's not applicable. Vaccine is not applicable.
No, sir. The thing is, that two things, the first, the immunity will be at the age when they during aging, no, the immune system will go down. Actually, vaccine ultimately it is eliciting our body's immunity to boost and combat actually. So naturally, aging, there will be something. And by age, people already, if they had sexual intercourse, they contacted the HPV, then the drug may not be very effective. That's one thing actually.
But no harm in having it.
I'm a scientist, so based on what I read, I'm telling you, sir. Some people they say vaccine they have causing some side effects. Okay, there's a big debate. Even for the COVID vaccine, whatever we all had on the debate. So the thing is, such a pandemic condition, no other people will die. Okay, there are millions of people dying, no other go. Okay, so that's what when balancing benefit and risk. Okay, so it has more benefit. Okay, so there are I know many people who have not taken the vaccine, but we know many people died. Who have not taken vaccine. So kind of such pandemic condition, always the government or clinicians who are they go for which has higher benefit than risk. Even there are little risk. Any drugs for that matter you take, see whatever chemicals you put in. For I'm telling you, like crocin, paracetamol, or acetaminophen, you take for longer time, it induces ulcer. In lab, we use paracetamol to induce stomach ulcer. Okay, every day we have started taking medicine. I would say up to 45 to year 42 years I used to take medicine all the time for everything. Little feverish I feel, kind of feverish, okay, we'll take paracetamol. Okay, little throat, kind of, even yesterday I was having kind of little throat. I was thinking, what to do. If I were 5 years ago I would have taken antibiotic yesterday night, but yesterday I had little nilavembu putting it. Okay, kind of little transformed to kind of traditional medicine actually. A little salt water gargling actually. I would say how I was like the time where for anything I take medicine in the sense like sneezing. Okay, little sneezing, or I feel like going to sneeze. Okay, antihistamine, cetirizine. Okay, I was taking like when I got married, my wife 10 years ago, she was, what this guy is like peanut taking, like eating peanut. I was taking medicine for everything. Then I never thought I would quit. Last 5 years, we haven't taken any medicine in the sense like not a tablet. Okay, really, sir. I never thought I would. The situation went like that. I'm taking no cetirizine for anything for sneezing. If I sneeze, if I take, it's okay. If someone sneeze, I will take medicine. Such a thing, overcautious. I very overcautious. Okay, my mind, sir, really it matters.
Vaccine in India. What is the name you said?
Gardasil, Gardasil, Gardasil.
Sir, any other vaccine or you have to only vaccine?
This is the primarily, sir, Gardasil and Gardasil 9. I think some Gardasil is the approved. Yeah. Very welcome, sir. Please, I would say, please try to avoid taking medication. So, so most of the time, body has the mechanism to combat actually. Number one, early diagnosis is the number thing. Or we have a lot of, let's food be the medicine I told, like if you have cold or anything, take like soup or pepper, we have all the ingredients. I'm telling you, like US, the Americans, they are appreciating all our Indian, spices and other things. I would say if you go to Indian, shops, first turmeric, raw turmeric. Okay, so see curcumin. Have you heard of curcumin? From the turmeric they isolated. It has a lot of property. There are 10,000 papers on that. It is anti microbial, anti all kind of thing. But if the trial failed, clinical trial faced because of poor absorption. Clinical phase trial, it failed. The reason is if you take isolate as a compound, if you take as a turmeric, it has a very good effect. That's why if you take our elders, like I would say, my father is healthier than me. His father is healthier than my father. The thing is, like our ancestors were healthy because they were taking all natural products and even whatever the food we had. Now we are kind of more attracted towards like Pepsi, Coke, all unhealthy junk food actually. Okay, that is only causing most of the issues actually. Kind of. So health, starts from the home actually. Okay, and I would personally won't take any medicine. I have made my friends, and I am kind of, my group of friends are like that. People who are our friends, they are smoking or taking alcohol. We see to that, we torture them like anything, they will quit actually. That's what we promote. See, we roam around, that's a different thing. But say no to alcohol, no to smoke. Character matters. I say you are a scientist or you are preaching those kind of things. Really, so first, that's very important. Okay, taking unnecessary medication, pain. You see, I never thought I can't take painkiller. Okay, there were situations actually for, and I'm kind of very weak guy in the sense. Now I'm healthy. That's a different thing. After stop taking tablets actually. The thing is, no, don't take unnecessarily. You will take for one purpose. Okay, like paracetamol. No, it's a, take for antipyretic and analgesic, whatever you take. No, so it is going to affect your liver and other part of the kidney. Kidney will be chutney if you take painkiller for longer time. Okay, it's well known actually. So please avoid. If the clinicians, the doctors advise strongly, but I know, like, now doctors are awesome, really, they say like only if it required, and Dr. Mohan, the world famous diabetologist, okay, he cares personalized medicine. He say like one drug, diabetologist, it cannot be given for. So he does meticulous, way of, treating the patients, and his talks, or even last week, we attended his Dr. Mohan's, international conference, is amazing doctors. There are excellent doctors as well. Okay. So now strictly you have to follow to what are the guidelines they give as per. Many people not take diabetes, they won't take medicine properly. They won't either they won't follow it properly or leave it, then say blame the doctor. Actually, it's not working. That's why I'm telling we have to appreciate the goodness from each medicine. It's not like Western medicine is good, tradition not bad, or the other way around. Okay, each and everything. Diagnosis, Western medicine, no doubt about that. For diagnosis, we have to use that. Appreciate. Okay, for cold and also a simple thing. Now you don't need to take antibiotics. In US also, and here also, I have seen like some of the good doctors. Even the doctor. Josephine from Isabel Hospital, she is a gynecology doctor from my wife when she delivered the baby. Unnecessarily she won't write, vitamins or bunch of, she won't write. So ethically, she say only you take fruits, eat a lot of fruits, this kind of thing. Okay, unnecessarily she won't give, antibiotics. Okay, there are people, a lot of wonderful doctors in India, with very good experience. Here, they see 100 patients a day. In US, only 10 patients they see. So experience wise, many people coming to India because of the expertise we have. Miyat Hospital, Ramachandra is amazing. We have, and you take wherever our Indians, proud to say, like Indians are very good knowledge and expertise actually when compared with other people. That's why in US lab, if you go, they take 2 Indian as a postdoctoral fellow, 1 or 2 Chinese. Okay, they are good at work. They will do, they don't, work on thinking too much, but they will. So that's how they run the lab. So we have wonderful minds here, the wonderful doctors. Right way of taking, will hopefully, the disease-free world in the future. Yeah. Excellent questions. Anybody else, sir? Yeah, please. Please free to ask. I am always tell, there is no question called wrong question. No question called wrong. Anything you can ask. Okay. Yeah, please.
So,
Hello.
so I just want to understand the healthcare ecosystem where the diagnosis plays the major role. So once diagnosis is done, then we can move forward for the treatment. So I just want to understand what is the practical challenges that we face in the initial phase of diagnosis in the real world. So for you to get a clear idea, I can explain a similar critical healthcare issue. Like cataract is one of the major healthcare issue, and to detect or diagnose the early stage cataracts, people are unable to access the healthcare systems. So for that, I and my team we are developing some AI-based cataract solutions for detecting it, like using mobile phones we used to.
Oh, nice.
So that is one of the approach where we made it as a two-step based diagnosis. So similarly, what kind of diagnosis systems do we have for this type of issues? And what are all the other ways in which technology can enhance the treatment measures?
Yes. Yeah, it's a nice question. So regarding diagnosis, as you said, early diagnosis for any disease, that really matters. And it depends on clinicians, So I'm a scientist, clinicians may throw more light on that. But for example, cervical cancer, okay, the pap smear is very commonly used. Any healthcare, wherever you go, So if some unusual vaginal bleeding or pain or anything, the women should immediately, go to the doctor, wherever, it doesn't matter, I mean, private practice or government practice, they will, simple pap smear, for sure, it will save life actually with respect to. But there are other technology, latest technology coming for other disease, for example, for above 40 years, they are doing diabetic test. Okay, so if there is sudden weight loss, okay, they will look for no whole body scanning to find out any lead that they get to for a particular disease for matter. With respect to cervical cancer, no, pap smear is the number one. So it most of the time, no, if there is any abnormality, for sure the doctor will intervene and treat that. Actually, that's the number one. And it's very interesting, like you doorstep service you are providing. So very interesting. I don't know how it goes for other kind of diseases. Maybe there should be a lot of implications taken from your work. So where do you work actually?
So I work with PwC. Before that, I was working with Tamil Nadu government.
Oh, okay.
Yeah. So there we had this e-pathway for cataract detection.
Okay. So we need much awareness. That's what people need awareness actually about each and every diseases, and you have to go and check that. And that's quite normal. Some people know the kind of over diagnosis also creates problem actually. Okay, so when you go multiple time, go and take chest X-ray, it's not advisable. But the doctors know usually they will recommend don't take too much of X-ray, unnecessarily, and it may have caused some issues actually. So diagnosis is very important, and over diagnosis is not good. Anything over, no, it's not good.
So, in your opinion,
Yes, sir. Sir, please, please.
between a carcinogen and cancer, because nowadays it is expanding like anything. Cancer is expanding like anything.
Yes, sir.
It's like lifestyle. For example, using detergent. Detergent is not a soap or top. It is a chemical.
Yes, sir. Yes, sir.
It's a dangerous chemical.
Yes, sir. Absolutely.
Surf everything. Tons, a million tons of surf is going down into the earth.
Yes, sir.
Not touching our bodies.
Exactly.
Between this and the cancer. Please.
Yes, sir. Say, for example, as you rightly mentioned, even few months ago, our government took stringent action on people selling this candy. Okay, in the beach, they sell candy. Okay, they use coloring agent. Okay, they were using, I think, rhodamine B, I think so. So rhodamine for coloring for the candy, so panjamutai, okay, for that they use. It's a cancer-causing agent. The children, they don't know actually. When they go to the beach, they will ask for, and parents will give that actually. In between, people are injecting formalin to prevent, the fish, from getting, rotten. Okay, it's a carcinogen. Okay, food. See how come, people. I would say like there is we need self-discipline. People who business is a different thing. Okay, you can't play the life of others actually. And I always tell like smoking. Like I told we have made many of our friends to quit smoking and taking alcohol. Okay, so there are some friends they have struggled a long time. Smokers for say 10 packets of cigarette they are not able to quit actually. But slowly they have given. What happens with especially with with alcohol, I can forgive 50%. But smokers, I won't forget for reason. Alcohol. Those who are taking alcohol, they are going to die. Okay, of course, their family they are going to affect it. That's okay. You are doing suicide. Smokers, second-hand smoke. Okay, you are not killing yourself. You are killing your wife, children, everyone. Actually, it's really sir. There is another counter called third-hand smoking. The people know a smoker is living in a apartment for several years, 10, 15 years or 20 years. He's leaving. Another guy is new. Is going the room. They are telling like, if you stay in the room for a longer time, you get affected. Okay. Now, e-cigarettes. Okay. The thing is, say, the problem with the cigarette is there are bunch of compounds actually. Okay. The people get addicted only to nicotine, but there are a lot of other compounds. That's a carcinogen that causes kill. Okay. But there are people nicotine based gums are there. People know. So I personally believe like if you have a strong mind, okay, you can do that. As you said, detergents, everything. Even earlier, people are telling people the baby using Johnson and I don't want to mention company name, but those very famous company in the powder, they say like it is a lot of causing a lot of issues in the baby. Using soap is giving some of the product they retracted, another they gave compensation. A lot of things are going on. So the best thing is I would say personally my opinion or my family, my wife or daughter, I have a 9-year-old daughter. We try to avoid using soap in the sense like you can use eco-friendly soap kind of thing. So for example, what is that? Handmade soaps. Okay, a lot of people are selling even toothpaste. Okay, last few years, 5 years, we stopped using tooth. Taste as such. use that powder, okay, herbal powder. It doesn't matter. It's not a specific brand. Anything without, because then the toothpaste they use sodium lauryl sulfate to get the froth. Okay, so it will, in addition to clearing thing, it will destroy all the the cells around your teeth actually. So we are living in a chemical world, sir. So as you rightly pointed, everything as here, it is going to the earth. Okay, the plants are growing there. It will take up those carcinogens. Again, the fruits what we eat, like life cycle, it's keep on. A lot of things are affecting that. I was talking about BPA and so BPA free bottle are there for kids. If you're going to bottle, they say BPA free bottle, but still it is a plastic bottle. Okay, it has other compound like phthalate based, like diethyl xyl phthalate. So in our department, all our professors worked on various, various organ system it affects. It is affecting. There are studies where BP and other these endocrine disrupting chemicals, EDCs, they're causing cancer. Why the sudden rise in fertility issues and cancer? It's all because of this detergent and other chemicals, sir. We try to avoid maximum possible things. So
shampoo is also a detergent.
yes, sir. Yes, sir. Really, sir. Avoid using that. You can use traditional shikai,
Hair dye.
Yeah, hair dye. Okay. Earlier, I was using some dye. Now I think most probably 1 or 2 years I may not need dye. Already I'm losing hair. But there are herbal dyes, which we get it actually. So, or we can go with many. I used to say before using dye, people say, oh, you're getting gray hairs. No, I say, salt and pepper style. I intentionally did, like anything is good. So we all are gifted with natural whatever available. So I personally don't like taking, artificial shampoos, perfumes, that too not on the body actually. No, it should not. It has alcohol content. Okay. Okay, those all are chemicals for sure. We will take for one purpose, it is going to affect some other purpose. That's how we are living a chemical world. Try to avoid using those kind of chemicals. Please avoid taking drugs without doctor's prescription, like over medication. It has become very common. Like this drug, we know everyone, because it's because of awareness. We know this. So please, antibiotics, please don't take antibiotics without the doctors, clinicians recommendation. And also if you say 3 days or 5 days, that only you have to take. Next week we'll have feverish, immediately we will take another course of that. It will make, I was talking about resistance, chemo resistance, drug resistance. We are developing resistance. And also when the manufacturing, the companies, the drugs, when you dump in the area, as you sir mentioned, like detergents, again, water gets contaminated, and taking those water, we will get, resistant to multi-drug resistance become very big issue, sir, actually. And we have to cooperate, not always all government or all doctors, all science. It's a, cumulative effect from all. Integration is required from each and self, discipline, and everyone should follow, like the rules and the, the signal follow. Each and everyone should follow the rules. Even no one is there in the road. If the red, we have to stop actually. But if we everyone makes together, we can really make disease free world, or even we can combat most of the diseases. Nice. Yes. Yeah. Very good.
Yes, sir.
Very good afternoon.
Just a general question. My close relation diagnosed with breast cancer.
Oh, sorry.
She undergone 8 cycles of chemotherapy. Now she's about to reach. So my question is whether in future any possibility of development of the same cancer cells. All these-
Okay. So very sorry to know, like your friend, she got breast cancer, but we can't tell that actually, So since she's taking medication, she may be 100% cured. If it is, if she is in, early stage, stage 3, unless it is metastasis, most of the people will be cured actually. Okay, but we can't know now. That's what doctor even know that proton therapy. One of my friends' nephew had a tumor actually. So here the doctor telling like it is reducing, it is decreasing, but we don't know for whom it will relapse. It may not relapse. There are people with cancer living for 100 years. Okay, we don't know. Okay, exactly, but hopefully, believe like she has taken chemotherapy and now on radiotherapy. hopefully, if it is not end stage, 100%, she will be fine actually. But exactly the doctors who is not treating her, they exactly know, oh, this is the stage. So she have better prognosis. So she may be cured actually. That's one aspect actually. That's like early stage means mostly 100%, I mean, yeah, it can be cured actually. That's what based on my experience. I'm a scientist, I'm not an oncologist, but based on the literature or what I heard from doctors, what I've read, so 100% it can be cured actually. The problem is the defense mechanism varies among. So say for example, if a group of people are there, we can't predict like people taking only 1 cigarette a day, 10 cigarettes a day. Based on, we can't say like people taking 10 cigarettes will get cancer first. We can't say that. All our buccal cells have no DNA repair enzymes. It differs from one person to person. So sometimes taking 1 cigarette, person may get cancer earlier to people getting 10 cigarettes a day. Okay, that's why asking not to smoke. Why one or two discussing? I said only one. I would say the same when I was in University of Oklahoma. One of my friend in the opposite lab, doctor, he is Yuting. His name is Yuting. So he was smoking. So being a friend, Chinese friend, I told, hey, Yuting, don't smoke. Please quit. No, he said, I don't know. I mean, you may get cancer or any of the heart issues. No, I said. He said, my father is 94. Sorry, my grandfather is 94 years old. He has been smoking for 40 years. He didn't get any heart attack or any cancer. No, why I'm going to get? He said, no, man, we can't predict. No, just like that, I was telling when I was you know, within 1 month he had a mild heart attack. Okay, he quit everything actually, but. We can't predict, no? We're based on one person. Oh, this guy doesn't have. He may have a lot of other for sure. He will have complications. Those who are smoking or having alcohol, because it is going to induce DNA damage or damage the cells. But it may not be, reflect in our system immediately. It may have some other. If there are ways like people taking alcohol, a lot of fertility issues. It has been well proven. Many things are well proven. Many things not yet proven. But whenever you take exogenous any agent, okay, too much of thing, okay, for sure it is going to have effect. And if it is early stage, 100% will be, and I'll pray for a speedy recovery. Yeah. Thank you.
So any further questions? One more question we can accommodate because time. Because we know that it's touching everybody's life. The subject is like that.
Good afternoon.
Good afternoon.
Question is regarding the treatment of cancer. As you compare the cell with the grown baby, can that cell alone not be removed from cancer? That cell can, if left, it can regenerate all the cells that you told. So can that cell alone not be removed to remove the whole cancer?
Yeah. Yeah, a very, very good question. Yeah. But practically speaking, the cells, locating the cells, see, you have cancer tissue, there are millions of cells. Exactly locating the cells or removing it difficult. But we can target it. So, say, for example, I've told a couple of siRNA based therapy at Cambridge. You can label that drug along, conjugate it with cancer stem cell marker we are talking like. So that is kind of no targeted therapy or medication where you can tag with another protein or antibodies which can go into the exactly bind with the cancer stem cells like the queen bee actually at this spot it can kill it actually. So we can have targeted therapy like only having the antibodies for cancer stem cells and conjugating our drug to that. Those kind of thing we can do. We can't just by remove it actually. But the thing is, if early stage of cancer, particularly the doctor will remove the tissue in such a way like no adjacent cells have any adjacents have any tumor. So they have a clear mark so that no other area is left with cancer. If such a case, most of the time all these cancer stem cells present inside the tumor will come off. Unless it is spread to the other thing, then again it will relapse actually. So otherwise, we can't just, as I said, take it out. It's very hard to. Maybe when you remove the part of the tissue, liver, one part of the tissue remove, along with that we can remove. But we can't take as cancer stem cells as such. It is not practically not possible. But use a drug or conjugate or label targeted only those cancer stem cells. Along with that, you can put those kind of drugs can be added, and specifically you can kill those kind of cells. That can be possible in the future. Very good questions from most of.
So thank you. Because of laxity of time, we know that a lot of questions will be there. This entire presentation will be, lecture will be available on YouTube also. So if you want to watch it several times, please do that. So on behalf of Madras Engineering Library, they want to honor the speaker with a pointer. So
Thank you, sir. Thank you.
then, founding member of TNSF for a short minutes.
Thank you, sir.
sir.
Thank you, sir. Thank you.
Excellent lecture. Absolutely free from taking enormous effort to be free from prejudices. I have to be as subjective as possible. Really remarkable quality for good scientist. I was just watching throughout the talk. It was an excellent demonstration of what is being scientific. Even within scientists, yes, sometimes they go their own prejudices, their own preferences. I thought that Dr. Ilangovan was taking very conscious effort to be free from any such prejudices. The question, I thought originally it was contained with research work. But even in the question and answer session, I was again and again watching him. Okay, never taking any extreme line, never asserting I am an expert or something like that. Okay, absolutely unpretentious. Really a good demonstration of what is being a scientific. Okay, thank you, sir.
Thank you, sir. Thank you, sir. It's a great
Okay,
blessings and honor.
excellent.
Thank you.
Stuff.
Thank you, sir.
Also thank the Anna Secretary Library Authority.